Thank you. We can open it up now to questions for all the panelists. So if all the speakers can maybe turn on their videos, and if anyone has questions overall, I see there's one in the Q&A from Sam Larson. I don't know if you want to ask the question, but I can, it's for Dr. Wisniewski. So you show the overlap between p-tau interactors and NFT proteins. Do you think the overlapping proteins could be important in aggregation and the non-overlapping p-tau interactors could be used as markers for tau prior to aggregation?
Indeed, we do think that that is the case. But again, we have a large number of proteins to examine and really ask that important question. So these are studies that are ongoing. And that's one of the nice things, but also challenging things about these proteomic studies. You identify numerous targets to follow up. And one has a plethora of choice as to what to study. But it's a nice problem to have.
Are there any other questions for the speakers from the talks?
So I have a quick question for Allison. So your work on platelets is very intriguing and exciting. Certainly platelets are the major source of systemic A beta. So have you been looking at any differences in platelet function, in particular activation state in Alzheimer's disease versus not?
I have to say I have not done that because I'm only really concentrating on, the amyloid properties, moving away from, where I would have done that, which would have been more if I were interested in the clotting and the atherosclerosis, I think.
Okay.
I think someone from the audience has a question. Nunzio Pamara.
Oh, can you hear me?
Yes.
Okay, Tom, I was very happy to listen to this program. I'm very excited. I have a question, actually. I have a general question, but mainly for Shane. In listening to his presentation, one gets the impression that astrocyte activation might be bad. We know that in the case of microglial activation, the experiments from Marco Colonna's group, as well as other experiments, have shown that in some instances, it can have beneficial effects, both in reducing toxicity from plaques, as well as decreasing brain amyloid burden. I would like Shane to comment whether there are instances in AD, in the context of AD, in which astrocyte activation may be beneficial, or is it always bad?
No, that's a really good question, Nunzio. And I have to say that the heterogeneity of both microglial and astrocytic responses is a burgeoning field for people to investigate. So the work that you talk about from Marco Colonna's lab is really interested because he knows that mutations in TREM2, which is an important phagocytic receptor, really change the pathogenesis and progression of AD in mouse models, and also the speed at which it progresses in particular cohorts of human patients. On the astrocytic side, I'm not even quite sure that killing a neuron is a bad thing, which I know is an outrageous thing to say, but evolutionarily, there is a reason for this to occur. And we actually originally thought that this was due to the fact that they were only killing neurons that were otherwise aberrantly firing and would disrupt the entire neural network, and removing them allowed the plasticity of the brain to rewire around that neuron-sized hole that had occurred. And we have evidence from this from work that we've done on prion infection and work that we've done in ALS as well. I will say that in our more recent studies in which we block only the neurotoxin, but we leave the reactivity exactly the same, the recovery from those in those animals is really impressive. So the other functions that these reactive astrocytes have, aside from the neurotoxin, is actually very beneficial for the recovery from these sorts of pathologies. You know, we know that they aggregate around plaques. We know that we need to remove plaques. We know that they actually then have the ability to improve synaptic connection throughout the brain as well. So I think it's a very, very complex system to start playing around with, but it's one that I think that we are slowly getting enough information to start doing that. The other piece of information I'll say in a tangential response to your question is, we know that ApoE is very, very highly expressed by astrocytes, more than any other cell in the CNS, and ApoE4 increases their ability to become these neurotoxic astrocytes compared to ApoE3 and 2. And this is some work that we had done with Dave Holtzman, and has also been done by Julia TCW and Alison Goetz lab just up the road at Mount Sinai. But if we look in non-industrialized populations in South America, there are a number of populations that actually have to be ApoE4 to make it through early childhood parasitic and viral infections. And so those populations then do not get Alzheimer's disease, but presumably have a large number of these otherwise neurotoxic astrocytes present in their brain, but they don't have the susceptibility on the neuronal side. So there's a nice lock and key to make this evolutionarily important. It just seems to be that in the Western populations that we're mostly studying, the susceptibility on the neuronal side becomes more of a problem. But really, really, really good question.
Shane, I think the complexity that you are describing perhaps might explain why anti-inflammatory agents have not worked in the treatment of Alzheimer's disease.
I agree wholeheartedly. I think the current treatments in that particular wedge of pharmacotherapeutic intervention drugs really go in there and they get rid of all of the functions and all of the subtypes. And so we suggest that you need to be a lot more specific and have a lot more fidelity in the drugs that you're using to treat those to treat these patients. So the glucagon-like-1 peptide receptor agonist that Alison also mentioned as well is a really good and very specific target. It really does play on the microglia ability to release these pro-inflammatory cytokines that does block only a small number of these reactive astrocyte subtypes, but it is still a number of them. And we're developing small molecules that will disrupt only the toxin production and release. So again, we're going to leave the reactive astrocytes there because we think that they're really important now and stop their ability to kill.
Thank you, Shane. Great.
I think we have one last question in the Q&A for Dr. Scharfman. Or actually 2 questions now for Dr. Scharfman. So Helen, do you want to address those questions? So there's one from Mitchell.
Sure. Yeah. So the EEG studies have done in other AD mice, such as transgenic for cow or triple transgenic express. Have they been done? Would the IS or seizures be stronger? Actually, a really good question. In terms of the mice, what's been done, a lot of EEG, but in terms of breaking down that EEG into the spikes, the seizures, and epileptiform activity that is neither, people haven't been done, in part because it's actually very difficult to do. You really need a number of electrodes. And one of the unanswered questions is, where are the seizures coming from? And are they coming from the areas where there's a lot of plaque? So early on in the TG2576 mouse, we see a lot in cortex. This isn't our lab that's first shown this by any means. But other people may not see that, other mice. And when you have the second, so question really needs to be addressed and has not. The studies that have been done, though, are a little bit daunting. So one study was done in an APP PS1 mouse by Heikko Tanila and Asla Pitkanen's group in Finland. And that was published several years ago in Journal of Neuroscience. And they saw in the same mouse, they looked at 3 weeks continuous video EEG with many electrodes, and the mice were very different, very, very different from one mouse to the next. Tremendous variability, which will mean that comparisons will be difficult to pull out statistical differences, but that doesn't mean that it shouldn't be done. Does not mean that at all. And so in that study where there are 2 mutations and there's more than 1 mutation, it wasn't as if there were more vigorous seizures or there were more than the many studies that have looked at 1 or different mutations. And when there's studies, though, that are done in some of the mice, it is notable that spontaneous seizures aren't found, but there's a high risk for seizure susceptibility. And since the seizures are rare, it's actually not clear that the people might not have missed a seizure. So there's really a lot of questions, and I think the answer is to do the EEG studies, do quite a bit of them, and try to get rigorous with definition of types of events, and try to get into the differences or the similarities. This is an excellent question. I'll just, for the sake of time, go on to the next question, which is, in AD patients, are the clinical severity and durations of IIS generally quite different? Well, this is another excellent question, and I actually didn't have time to address it, but I thought a lot about Thomas's talk, because that those rapidly progressing patients might actually be people who have quite a bit of seizures, and I was actually going to ask about that. Sometimes the very vigorous seizures occur early in familial AD, and those individuals quite tragically progress rapidly. But I'm not so sure that that applies to Thomas's experience and his data set. In terms of what's being done now, is a lot of researchers have studied and are continuing to study individuals before the diagnosis of cognitive disturbance, and also later. And there's a very complex data that are emerging from that. I don't think they are the be all and the end all yet for the difficulties that I already mentioned, but they are showing that the abnormalities can occur very early. And beyond that, it's very, very hard to address specifically the interictal spikes. They're going to need continued investigation.
So like Dr. Sharfman mentioned, for the sake of time, we have one last question. I'm going to allow Dr. Goni to talk. Can you speak?
Can you hear me?
Yes.
Yeah, it's a question for Alison. For what she said and for what we know, most probably during life, we'll have like thousands of times more A beta in circulation than in the brain, and because of the platelets. However, we don't have any report that outside the brain it will change conformation. That is the only problem. When you change conformation, you have pathology. If you don't change conformation, you don't have pathology. So you can have all the beta that you want, but if you don't change the conformation, and that is happening only in the brain. So I wanted to know if it is possible that with the system that she has and is developing, you can study what is the difference and what is bad or good in one place or the other that will lead to pathology or nothing.
You know, I agree. You can't replicate it perfectly, but we certainly can make oligomers and do other kinds of things to change the conformation of the amyloid. And we have done work like that in our lab. But then, you know, you are adding another artificial layer on. But you're obviously, that is correct. Again, any model that you make is only a model. I can't get in and do a real brain, and I don't have the 30 years. So we have to do as we can, and again, build it and improve it and refine it as we go along. But I mean, you obviously make an excellent point.